Today I'm going to be talking about our lab's efforts to sequence H5N1 out of grocery store milk. So first things first, what is influenza? We all know it's like, a cough, a sore throat, and a fever, but it is a virus as well. It's an RNA virus, single-stranded, and it has 8 gene segments in it that encode for 1 or 2 proteins each. Huge genome, 800 base pairs to 2.4 kb. So I'm scared of all of you with your plants and animals. On the outside of the viral envelope, it's decorated with HA and NA receptor proteins, and that's where we get the H number, N number, H1N1, H5N1. There's a whole bunch of combinations of those that you can get. Today I'll be talking about H5N1, and then if you're really into the weeds, specifically clade 2.3.4.4b, which is the HA type. So bird flu is native to birds. I know this is big news. It's got a natural reservoir host in wild birds, specifically waterfowl and shorebirds, which conveniently are some of the biggest migrating species on the planet. So what happens in bird flu in one place doesn't stay there. It tends to kind of be spread and to drift on these migratory pathways. These are just sort of broad strokes, but they're all kind of connected. We live in a world that's awash in birds and therefore bird flu. So this specific worrisome H5N1 strain was first identified over in East Asia before 2015 or so. And from that point on, it's sort of taken a westward route along these migration routes, passing from bird to bird into Western Europe in 2017, where it did a ton of damage to poultry industries, wild birds, and then also spilled over into mammals and did some mammal-to-mammal transmission at a mink farm, that's worrisome because if you're transmitting bird flu in mammals, potentially it picks up adaptations. It learns how to be good at transmitting in mammals, which is something that I, as a mammal, don't like. So then after 2022 and 2021, 2022, this virus once again caught a ride on some wild birds and made it to the Americas, where it's caused massive impacts to wild birds on these continents, in addition to a couple concerning mammalian outbreaks again. In South America, there's a huge outbreak in marine mammals, so lots of seal die-offs and ongoing transmission in that population. And then last year, there was a spillover event into dairy cattle in the US of A. And so this outbreak was identified in spring last year, and then really quickly after the news broke, the FDA came out and said, we found viral RNA in milk. And so we took that as a challenge. We went out and we bought a ton of milk. And we said, I bet we can find RNA in this milk. And spoiler alert, we could. But I just wanted to talk about what the process of milk, what the supply chain looks like, to kind of get a better idea of what our sampling strategy ends up looking like. So milk starts with a cow. You milk a cow. You do that 2 or 3 times a day. You pool the milk from all your cows on your dairy farm into a bulk tank that is somewhere on the premise. And then once a day, sometimes twice a day if you're really going crazy, that bulk tank gets transferred to a milk hauler that takes it away to a dairy processing plant. And the dairy processing plant pools it with the other farms around, and that's the stage where you get homogenization, you get pasteurization, and you get packaging as well. And then the retail milk that we see on the store shelves actually has a plant code on those cartons. So this is my big takeaway. If you go buy milk, you should definitely look for the plant code on it, and you'll be able to see what exact processing plant it was processed in. So the website is whereismymilkfrom.com. So far, we have purchased about 256 cartons. I bought some here, so that number will be going up. And we've tested them for H5 with a qPCR, and we've gotten 43 of them that were positive. Of those 43 positives, we've gotten 10 cartons with really good sequence. We're working on getting that number up, but we're defining good sequence here as over 90% coverage across the genome at 20x depth. And we're using a tiled-amplicon approach for this, which is perhaps a bit strange in flu, because flu is actually very neat in that all 8 of those gene segments have ends that are basically conserved. So you can actually design a PCR that amplifies every segment from the genome all at once, which is super great, gives you a ton of information, easy to sequence. However, we noticed that that doesn't work well for our samples. We think that's because when you homogenize milk, when you heat it up for pasteurization, you're breaking down some of the RNA into smaller fragments, getting some shear. And so a tiled-amplicon approach that's a bit smaller enables us to just amplify little tiny bits that are probably still there in high numbers and get across those like breaks where it would cause a normal PCR to fail. And so a lot of this work is sort of just a redirection of our lab's work on COVID sequencing. We do a ton of that, and so we were like, well, we can sequence flu too. So we designed that tiled-amplicon scheme using primal scheme, which is a very cool tool that essentially just pumps out a tiled-amplicon scheme for you, given some reference sequences that you're interested in. And then we literally just substituted that straight into our workflows, did our normal reverse transcription, amplification off of our milk RNA, and then we sequenced using Oxford Nanopore Minions. And this protocol is up on protocols.io if you are interested in doing it yourself. So moving into the outcomes of our sequencing, we are pretty happy with the depth of coverage that we're getting here. This is a coverage plot showing position on the genome for the segment PB2, and then all of the colorful lines are different cartons that we've gotten. And then on the y-axis, you've got depth of coverage on a log scale. So you can see that when we have an amplicon covering a region that works, it really works. We're getting pretty outrageously deep coverage. Probably doesn't need to be that deep. We'll optimize it later. Viruses move pretty quick, so we try to do the same. But we do have some amplicons that are dropping out. That's where those lines kind of drop below this sort of cutoff line. That's a 20x coverage cutoff line. And I'm working on optimizing those. Don't have any results that I have done that yet, but I'm working on it. And so when we zoom out from just looking at that one segment to looking at all 8, we see a very similar pattern. We're getting a couple amplicons that drop out, but overall, these are all above 90% coverage. It's pretty good. We're pretty confident that these are useful for looking at individual mutations at enough sites on the genome that it's useful, it's informative. So when I say looking for mutations, what do I mean? In the context of flu, there are a couple things that we're watching out for that would turn bird flu into human pandemic flu. And those 3 things broadly are mutations in the HA that would increase respiratory stability, so its ability to exist in the air for long enough to get from me to you. And then mutations in HA again that can increase receptor binding, so it can actually infect mammalian cells more effectively. And then once it's inside those cells, we're looking for mutations that utilize mammalian replication machinery more effectively in the polymerase. So if you end up in the middle of this Venn diagram, you're going to have a bad time. So we have a little short list of mutations that we think are really involved in these, and we kind of go through and see if we find any of them. And out of this short list, we've detected 2 out of the 6. But maybe don't panic quite yet. I'll talk through them. So the first one that we detect is in HA. It's P324L. We've detected it, but we only detected it at subconsensus in one of our sequences. So hopefully we don't detect it again. flu is kind of random. This is a virus. It's pumping out so many variants. So we're hoping that doesn't, increase in frequency as we sample and get more sequences in the future. And then the other really concerning mutation that we see is PB2, M631L, but that's not really novel. We've known about that because every single flu virus from this cattle outbreak shares that mutation pretty much. So we're concerned, but this isn't exactly a sentinel warning about that. We already knew that was something to be worried about. So importantly, we're not seeing E627K. That's kind of like the biggest, baddest one that people lose the most sleep over. What are we detecting? We are picking up mutations. So that's the point of this slide. The details of these mutations don't matter as far as we are aware. If you're a flu expert, you probably know more things than I do because we just started this. But we're picking a lot of these mutations up in 1 or 2 samples, not necessarily broad throughout all of the sampling that we're doing, and not necessarily in a way that's increasing over time, which would be, a sign that maybe there's a selective pressure acting to increase the transmissibility of something. So where do we want to take this research next? We think that it's really important to do increased testing in dairy cattle, and then sequencing along with that, because why not? It's really easy to sequence once you've tested. For us, that means continuing to buy milk and test it and sequence from it. Sadly, we've been having a harder time finding milk with flu in it, which is actually great, but I think it's probably because we don't buy our milk in California. So I've tried to grab some samples from here to take back and test. So in Wisconsin, we've been testing a lot of like yogurt, ice cream. We're getting desperate. And so yogurt and ice cream have multiple ingredients. This is an ingredients list for eggnog. There's dry milk in there. There's cream in there. There's whey in there. So we want to be talking to processors when we get a positive like this to say, okay, would we test your fresh milk? Could we test your pile of dried milk that maybe is old, that's from like Colorado last year, so it makes sense that there's bird flu in it? Or is what we're observing in your fresh milk? Just so we can like kind of get a better understanding of the local situation. And then for the U.S. as a whole, obviously I'm not making any policy recommendations or anything, but the USDA is working on getting more bulk tank testing. So that's on-farm testing of that initial pooling. That would be super powerful for getting more visibility into this, doing more action to stop it. We cannot encourage them to do that enough. So good job, USDA, and please bring bulk tank testing everywhere, especially to Wisconsin. So I would just like to thank everybody in the O'Connor lab for helping out with this project, especially Dave, who cooked up this crazy idea to sequence out of milk, and then Nick Miner, who did all of the bioinformatics from putting together like the Nextflow pipeline to run from base calling to those pretty figures that I shared. And then also I'd like to thank our undergrads for being absolutely amazing at buying milk. They found so many plant codes that we had no idea about. And then this is available as a preprint as well. So yay, check that out if you're interested, and I'd be happy to take any questions.